Drug use for medicinal and recreational purposes has been taking place for almost as long as humans have walked the earth. Morphine was the first non-alcoholic addictive substance to be manufactured and widely used. In 1806, a German pharmacist revolutionized pain control by extracting crude opium. By the mid-1800s, however, researchers began to realize that opiates were addictive. In the wake of the American Civil War, thousands of veterans became addicted to pain medications such as morphine and opium. As late as the 1890s, one could buy a syringe and a small amount of cocaine from the Sears and Roebuck catalog for $1.50. And even Coca-Cola had trace amounts of the drug in its patent medicine soda. By the beginning of the 20th century, the global trade of drugs was becoming a problem that required a global solution. In 1909, the U.S. passed the first federal ban on the use of a non-medical substance. But many drugs, including opiates, could still be used medicinally. At that time, we lacked the science to properly understand addiction. The first drug tests were not the technological innovations we see today. Drug use and abuse would not be well understood until tools and technology were developed to measure the body's biological response. Chemists tested the saliva of racetrack horses suspected of doping by force-feeding it to frogs. If the frogs reacted erratically, the drug test was considered positive. This gave birth to the first drug testing laboratories conducting horse saliva tests. However, in the 1950s, chromatography emerged as the preferred technique for drug testing. The first immunoassay was developed by Burson and Yalow in 1959 and resulted in a Nobel Prize in Medicine. The development of this technology gave birth to a new company called SIVA, responsible for the reliable drug testing that fuels our court, military, and medical systems today. The next years brought a boom of not only innovation in drug testing, but also a war on drugs that would last decades.